All right. Hey, everyone, and welcome to the second webinar on the MNE toolbox. Very quickly, in the previous webinar, we looked at some of the basics of the MNE toolbox. Rob took the example of NIRx data and showed us how to load, visualize the data, as well as walked us through some of the pre-processing steps. And we did some statistics in the end, not too much though. In the second part, Rob will do a deep dive, if I may say, and talk about GLM as well as group level analysis. And we at NIRx like supporting our users, not only through the technology that we build, but also making integrations with cool technologies such as the MNE toolbox, which are outside our ecosystems seamlessly easy. And before I give the mic to Rob, let me introduce my team, the team behind this webinar. I am Mahipal. I lead the scientific consultant team at our Berlin office. With me is my colleague, Amy from marketing, working very hard to put everything together, schedule everything, and send you those invites. Alina is also joining us in the background. She will be taking your questions, putting them in a nice way so that we can ask Rob those questions. Before we move on, a few practical considerations. Of course, we like to hear from you. If you have any questions, please write them in the questions panel of the GoToWebinar. For this webinar, everyone will be muted. As usual, all our content, all the previous webinars, including this webinar, will be recorded and it will be available on our website for future reference. And of course, if you are not able to answer any of your questions, if you run out of time, or if you happen to have any questions later on, you can always write to us and we'll be very happy to answer those questions. So without further delay, let me introduce Rob very quickly in case some people did not join the previous webinar. Rob is a researcher at the Department of Linguistics at Macquarie University in Australia. At the moment, he's in Melbourne, past midnight his time. So Rob, we are always grateful for that. He's a PhD in cognitive neuroscience, and his stint with fNIRS started when he started working with patients with cochlear implants. So I won't take too much of your time, and Rob, floor is yours.
All right, thank you. So you can see my screen?
Yes.
It's not full screen.
Great.
But
not full screen. Let's try full screen. There we go. So thanks for the introduction. Thanks for having me today. This is the second webinar, and we'll follow a similar format to the first one. We'll start with some questions, and then we'll take a quick look at the documentation and how you can get help, and then we'll move on to an actual GLM analysis. I'll go out of full screen mode. I don't think it's. So here are some questions from the seminar registration form, and I thought I'd just address them up front so that you can think of more questions throughout the talk. So one of the questions was, is batch processing possible? So yes, and we will get to that today. I will move faster so we don't miss it. And then another question is about different fitting paradigms like autoregressive models. Yes, we will also address that today. So remind me if I forget. And we will, but yeah, in short, you specify the noise model to do that. Similarly, with exporting GLM data, we will talk about that today. Someone asked a question about converting coordinates to MNI positions. We won't talk about that today, but the functionality does exist, and you should go to head to MNI. So that's where you would find that, and ask on the discourse forum if you have any troubles. And somebody also asked about blind source separation. I don't think we'll get to that today, but again, please feel free to ask on the the help forum. I also got some questions and some feedback online from the previous talk. So one person said it was confusing to have two sets of documentation. And so what I've done is I've updated the MNE NIRs website based on your feedback. And so you can now find, this is the MNE NIRs website. If you click on API, I've now included all of the functions from both MNE Python and MNE NIRs together. So hopefully that will make it easier. Thanks for the feedback. Somebody also asked about how to load their montage, and maybe I skipped over this too quickly on the first talk, but for NIRx data, the montage will be loaded automatically if you use the read raw NIRx function. So you don't need to load a montage on top of that. The information is automatically loaded, but I'll show you today how to validate and make sure that it was loaded correctly. And somebody also asked about renaming and removing triggers, and we'll address that today. So today I'm going to go through, I showed this sort of figure last time about what MNE is, sort of packages these existing tools. And today we'll use a lot of the functions from NILearn, which is an fMRI toolbox, and they have a lot of GLM functionality, which we've repackaged. So if you need further information, I'd recommend also looking there. And I should also say that a lot of this has been built by people other than me. And so make sure to, sort of cite these other packages accordingly. And yeah, I think that's probably the introduction. So today we're going to work through a GLM analysis on the same data set as last time. So hopefully we'll get a similar result. And then we'll move on to how to do a sort of batch or a group level analysis. And just like before, I might do some funny things just to simulate, demonstrate some functionality that this toolbox has. And please ask questions throughout, because it makes it a bit more interesting. So if you want to follow along today, you can go to the MNE NIRs website. And if you click on the examples, what we're going to go through today is the GLM analysis toolbox example, and then we will go through the group level analysis demonstration. And so if you click on this, if you want to follow along today, if you go to the bottom and you hit this launch binder, then it will temporarily not work. Well, there you go. All right, so don't do that. We'll just follow along with me today. All right, so they're the two tutorials which I'll run through a modified version of. So like last time, we're going to work through a notebook, which you can see on the screen. And so we start off by loading the packages that we need. So we load the MNE and the MNE NIRs package. And we'll also load some extra plotting functions from NILearn, which I'll show you later. And the data that we'll analyze today is data recorded on me, and there's three conditions. So one where I'm tapping with my left hand, one where I'm tapping with my right hand, and one where I'm doing nothing. And I tap for 5 seconds, and there's a random interstimulus interval of, I think it was maybe 15 to 25 seconds between each condition. And so what we would expect, or what we hope to see, is that when I'm tapping with my left hand, there's activity on the right motor cortex, and when I tap on my right hand, there's activity on my left motor cortex. So that's what we're sort of aiming for. So once we've loaded the packages, the first thing we'll do is load our data. And so this here, I'm just using a standard example data set, and that lives in this fNIRS raw directory. And so we're going to load that with the mne.io.readRaw NIRx, just the same as the first talk, so I'm not going to talk about that. And then we're just going to downsample it to 0.7 hertz, just to make this example run a little bit faster. So when we load our data, just like last time, it gives us an automatic summary of the data. So we can see that there's 56 channels, the recording was 50 minutes long, and it, as we requested, it's been downsampled to 0.7 hertz. And so yes, I got a few questions through different mediums after the last seminar about how to load their montage. So all you have to do was use the redraw NIRx, and that's loaded the montage, and you can check that by going raw intensity.plot sensors. And so here we can see that this is the montage, and the sensors are located over the 2 motor cortex regions. And so the more advanced example is you can use this MNE.vis.plot alignment, and the exact arguments are in the example, which I won't go through now. But if you use this MNE.vis.plot alignment, it will show you the positions in 3-dimensional space. So this can be handy to ensure that it's been loaded correctly and that the sensors are where you expect them to be. And actually on top of that, what we can do is we can even, let's say you're interested in the precentral gyrus on the left and the right hemisphere, we could tag those in green so that now we can ensure that our sensors are indeed over the correct location. So that's just a nice way to validate that your montage has been loaded correctly. But now I'll move on to the actual point of this talk today, which is the GLM analysis. And so once we've loaded our data, the first thing you usually want to do is convert your triggers into a sort of more meaningful name. And I think I moved over this too quick in the last talk because there was a follow-up question about it. So you would use raw intensity dot annotations dot rename, and then you pass in that trigger 1 means control, trigger 2 is tapping left, and trigger 3 is tapping right. I also normally have a trigger at the beginning and the end of the experiments, just so I can cut the data to the correct length. And we're not going to use those for analysis in this particular example. So to remove an annotation or a trigger, use raw intensity.annotations.delete, and then we delete to 15. And the last thing that I normally do is tell the software how long did the condition last for. So I was tapping my hand for 5 seconds, and so we would set the duration to 5 seconds. So if we run that, we can see that there's now 90 segments, 30 control, 30 tapping left, and there'll be 30 tapping right. That's cut off. And we can check this, and we can check that the triggers were randomly ordered. It's the same as last time. And so this is a plot with time along the bottom and the event along here, and you can see everything's randomly ordered. I'm just moving quickly because this was discussed last time. And another way to view the trigger events is to plot them as a boxcar. So we can ask for the create boxcar function and pass it our raw intensity data, and it will create this boxcar function. And that can be handy. And you might want to just look at the first 30 seconds, for example. So you could maybe look at the first 130 seconds. And you can see that the boxcar functions are as you would expect. So they're up for about 5 seconds and they come back down. And so normally what we would do then is, so we have our raw intensity data, we would convert it to optical density and then apply the Beer-Lambert law. So that's the next step. So now we have what we call like the hemodynamic signal. And what we're going to do first is split that into the long channels and the short channels. So there's 2 convenient functions here, get long channels and get short channels, I think. And so if I run that, we now have 2 variables. One variable called short channels, which has 16 fNIRS channels. So there's 8 short channels and there's 2 light frequencies for each one. Oh, sorry, I should say there's 8 source detector pairs and 2 light frequencies. And for the raw long channels, we now have 40 long channels. And so the next step is, so now we know when our triggers are, we have our data, and we also know the duration of our stimulus, and we can combine that information with a hemodynamic model to create a design matrix of the sort of a model response. And so we use the function make first level design matrix, and that takes in the raw data, and you specify what type of HRF model you would like to use, the duration of your stimulus, and the type of drift model that you would like to use. So we'll use these parameters to start with, and I'll show you what that creates. So that creates this design matrix, and we've plotted that using the plot design matrix function. And along here, along the x-axis, we have the different conditions. So we have control, the tapping left and the tapping right. And going down the screen, we have time or sample number, or it's called scan number. That's a hangover from the fMRI world. And so this is the model that will be fitted to our data. We also fit a constant. So if there was a DC offset in your signal, that would account for that. And then we also have drifts, and you can specify the drifts that you want. So this drift here, it's just an upwards drift, and that might be something like if the machine was warming up, or something was steadily increasing throughout the measurement, this would account for that. So this is a design matrix that we're going to use later. We can also include short channel regressors in this design matrix. So here I'm just going to add 2 extra regressors to the design matrix, one for the oxy and one for the deoxy signal. I'm just going to take the average of the short channels to do that. So I specify that as in I go design matrix, short HVO, and then I just set the mean of the short channels. And so if I do that, we can now see that the design matrix has 2 additional columns that account for the 2 short channel types. And you could also add more columns for the PCA components of the short channels, or you could do all the individual short channels, but this is just an example. And the last thing I wanted to show was how to add another column for some magic regressor that you might have. So if you have some super secret predictor of neural responses, then you could add that as well. So here we just add some random noise. So this, and I call it the magic regressor. And so we wouldn't really expect, because I've just added noise, that this would explain much of the neural signal. It's just an example of how you would add another column. So that's how you have your design matrix. So you have the data, and we now have the design matrix, which is meant to represent the expected model of that data. And this can be a bit abstract, I think, this sort of 2D plot. So what we can do is we can actually just, let's look down this first tapping left column here, and we can just plot it. So if we plot that first column, what that looks like is this. So you've got your stimulus, we know what time the stimulus occurs, and this is the model that we'll be fitting, the model response. All right, so we're just going to start off by fitting a GLM to 2 channels to keep it nice and simple. So I'm just going to make a data subset by taking the raw data, making a copy, and just picking the first 2 channels. And if we would like to run a GLM, then at a minimum, what we need to do is the data, and we need the design matrix, and we run the GLM on those 2 things to get estimate. So if we run that, we get this variable called GLM est, or estimate. And if we look at that, that's just GLM results for 2 channels. And so there was some advanced questions. I think if we go back to I've lost my questions. But so people asked about the model and autoregressive models and sort of these more advanced questions. And so this here is a class which stores the GLM results, and it provides some convenient functions. So I'm not sure I understand. Well, Siri is talking to me. And so you could just ask it for the channel name, something simple like that. And just like in the other types of data, you can ask it for just the HBO or just the HBR by using this pick function. So if we wanted just the HBO channels, it would return just one channel. But perhaps you would like to dig into the details. And so you can also, it has all the information that you would expect from a GLM model. So you can look at the degrees of freedom of the model, the residuals, the R-squared value of the model, or what was I going to show here? So by default, the GLM will use a first order autoregressive model. And so if you want to look at the models, you would say, for this channel, give me the model. And you could ask for, this is all the information. So you can get the order of the model. So we use the first order model. If you would like to use a higher order noise model, you have to specify that in the GLM. So you would go noise model equals, so you can do ordinary least squares, or you could do a fourth order autoregressive model. So if you want to use a higher order, you go AR for autoregressive, the number that you would like to use. So, sorry, the order. So to run a fourth order autoregressive model, we can now see that the model order was 4. And if you would like to look at the fitted autoregressive parameters, you would request, it's called row. So this is the autoregressive parameters here. It's advanced, but it was a question that we received, so I thought I'd dig into it. And somebody asked, how would you get the whitened signal, or how would you check it was whitened? I can't remember the question. So here's an example of how you would plot. So Y is the input data. We also have access to the whitened Y. So if we want to plot those two together, we can see that the input signal is kind of all over the place, but the whitened signal, I mean, it looks a bit whiter. Whereas if you did a ordinary least squares fit, it doesn't have a row, that makes sense. Then it doesn't, there's been no autoregressive fit here. So that's how you do the autoregressive fits. That's all quite advanced. I think what most people would use is the convenient functions that we've sort of laid on top. So you've done your GLM, you run your GLM on your data, and now you would use this toDataFrame function, and it's going to produce for every channel, for every condition, it's going to produce an estimate of the response, and you get the p-value and the standard error in a nice table. And it will also neatly sort of deconstruct the channel name into source, detector, and chroma, so that you can easily filter your data later. And if we just look, so sometimes these tables can be really long. So what I like to do is I just like to look at the first 10 items. And so if we look at this, we can see that our control condition, there was no significant response. The magic regressor, which was just noise, also had no response, which we expect, whereas the two tapping conditions did. So in this particular channel, we can see that the results seem to make sense. And another convenient function is, I mean, looking at these tables can hurt your eyes. So there's convenient plotting functions. So if you just call .scatter, it will plot this in a convenient way. So here we have the GLM estimate on the y-axis, which we call theta. I think other toolboxes call it beta. And we can see that the control estimates and the magic regressor are close to 0, whereas the two tapping conditions have a positive oxyhemoglobin response and a negative deoxy, as we would expect. So are there any questions coming to this point, or can I push on?
There is one, Rob, indeed. It's related to having regressors in GLM. And I'll read the question to you. When collecting data with the NIRx NIRS-2 system, there's the possibility to include an accelerometer to assess movement as a further potential regressor for artifact correction. Is this accelerometer sensor data already accessible in the MNE object? when using the .snrf data structure?
It's in progress. So at the moment, it just reads in the data. I'd hope in a couple of weeks that it would also read in the accelerometer data. If you read in the accelerometer data in a separate step, then you would use sort of something like my magic regressor here. So you would just say like accelerometer XLX or something, and you would, if you've read it in with, you would pass in, your variable here. So at the moment, no, I haven't programmed in a reader for the accelerometer, so I'll add that to the to-do list. Actually, that's a good point. Is any features that are missing, please let me know, because there's sort of no end to the things that I could add, and I sort of it's hard to know what's useful to people, and so if people can sort of, if you go looking for something and you can't find it, don't give up. Just sometimes it's there, but it's just not publicly exposed. So just put a little note in the issues on the website, and usually it's pretty easy to add. So so that's something that we can add. Mm-hmm. So we'll move on. So that's how you would analyze just one or two channels. But let's now analyze the whole data set. So we'll just do the same thing as before. We'll run our GLM, but now on the entire data set with the same design matrix. And we can see that we have GLM results now for 40 channels. And now you wouldn't want to plot 40 channels in a scatter plot because it'll be a bit of a mess. So what we can do instead is plot a topographic representation of the results. And so we would use this plot topo function, and we would tell it which conditions we want in the topographic map. And what that's going to do is plot on the top axis is the oxyhemoglobin responses from the GLM, and on the bottom axis is the deoxyhemoglobin response. And we can see that when you're tapping with the left hand, the response is larger on the right side, and when you're tapping with the right hand, the response is larger on the left side, as would be expected, and that the oxy and the deoxy are kind of going opposite directions. Perhaps you would want to plot the control condition as well to demonstrate that there's no response during the control. So you can just pass in as many conditions as you want. So you might want the left and the right and the control, and it will make 3 plots. So that's kind of a convenient way to plot your data. What I don't like about these plots, so here you have the little dots are where your channels are. We have no measurements in the middle of the head here, but it's interpolating between those points. And I mean, it's fine if you know what you're looking at, but I don't think it's very intuitive. So what you can also do is just plot each section individually. So here you can see that the data is smooth across hemispheres. I think it's a little clearer, a little more honest to say that we only had sensors here and we only had sensors here. And we do that by taking the GLM results, making a copy, just picking the first 10 channels and running the plot topo on that. And then in the second, so that would generate this left side, and then to generate the right side, take the 10th to the 20th channel, and we plot it here. So that's how you can make, I would say, a bit more honest plots. And another way, this is a nice way to represent your data, but it's another way you could do it is by plotting the results on a head. So so another thing we can do is, what I'll do is I'll plot the tapping with the left hand. And so what we've done is we've created the same head that we had originally when I was showing you the source detector pairs. And now everywhere there is a line, that's a channel. So there's a source and a detector. And the color in between that bar is the size, is the GLM theta estimate. And what I've done is I've colored in green the part of the brain that you would expect to be active. And we can see that, so for this one, when I was tapping with the left hand, the right hemisphere has clearly a larger response than the left. And you can see that it's sitting, the largest part of the response is sitting right over the brain region that we would expect. So this is another way to visualize the same data. So this is all being the same data, but just plotted in different ways. So that uses the plot NIRs source detector function. And then the adding the green labels is a bit of a fancy extra. So you use this add label function. Now, what we've also been working on, this is a bit of a sneak peek preview, it should be finished in the next week or two, is surface projections. So what you can also do is project the estimates onto the nearest cortical surface. So this isn't a proper photon migration. This is just a way of taking the closest surface to each channel and projecting the response there. But it's the first step on the way to a proper photon migration analysis. But I think it still helps to represent that most of the activity was on the right side of the brain. So there are 3 or 4 different ways that you can look at your channel level data. Now I'll move on to how you would do your sort of region of interest analysis with the fNIRs. Unless there was any questions that have come in before now. Nope.
Not at the moment. No.
Good. Okay, so before you started your experiment, you picked a region of interest to analyze, and you picked your sources and detectors that would be in that region of interest. And that's sort of what we'll analyze now. So in this experiment, we're going to split the data, all the channels, into channels on the left side of the head and channels on the right side of the head. So you would pick pairs. So you need to create a list of pairs that you're interested in. So we're going to have one set of pairs, which is source 1, detector 1, source 1, detector 2, source 1, detector 3, source 2, detector 1, and so on. And we're going to assign that to the left side, or the left region of interest. And then you pick another set of pairs to assign to the right region of interest. So if we do that, and then we need to, so now we've got these lists of pairs. We want to find out what index that is within the MNE structure. So we use this pairs to index function, and we pass it the data plus our desired list of pairs. So once we run that, now we have a list of groups. So we have a left region of interest group and a right region of interest group. And I can show you what that looks like. So now in the left region of interest, there's all these channel indexes, and on the right region of interest, there's all these channel indexes. And we also would like to look at the conditions. We're only interested in the control and the tapping left and the tapping right for this example, but we're not going to look at the extra regressors. And so then we take our GLM estimate and we ask it to give us a data frame for the regions of interest, and we pass in our group, so our group of channels, that's our regions of interest, and we pass in the conditions which we are interested in. So this will calculate the responses for the region of interest. So now we have a data frame which contains, instead of channels, it's going to contain for each chroma, for each condition, so we've got control on the 2 tappings, and for each region of interest, we now have an estimate. And I'm old-fashioned, I like a p-value, so I'll make an extra column that has p-values and decided if it's significant or not. So we can see that for the control condition in both regions of interest, there was no significant response, whereas for the tapping in the left side and the tapping on the right side, both regions of interest were significant. And so that's how you would get your sort of your summary level. This is your individual participant region of interest GLM results, and then you might want to export this for analysis in your favorite analysis program. So this is already a data frame, so you could just go to CSV, and you could save that as my results.csv. And so now if I look here, I now have my results.csv, and that's been saved in the CSV file, which you can use with your favorite program. I think that's where I'll, I think that's where I'll stop the individual level analysis. Unless I've missed anything, Mayapol, is there anything that springs to mind?
We do have a couple of questions that we may take.
Perfect.
Seems relevant to this part. And I'll start with the one on GLM. I think you covered this to some extent. Is it possible to use different GLM fitting paradigms like whitening of residuals or autoregressive models? And follow-up question on that is, how can we export the GLM data?
Okay, yep. So I showed how to export the, this was the region of interest level data, but you would use the same technique to export the channel level data. So here we have the GLM, let's go. Yeah, so like I showed before, we have the GLM estimates. We have 40 channels here. So we could convert that to a data frame. And that's this data frame, which I showed before, which has all the channels. The same way you would go to CSV and you would say sort of my channel level results.csv, and it will save that. Regarding the autoregressive models and the whitening, so yeah, we briefly touched on that. Sorry to make everyone dizzy. So you specify that by using the noise model parameter here. And if you use an autoregressive model, then it will whiten the signal, and you have access to the whitened signal as this whitened Y. And so there's a lot more details. If you do dig in to these models, it gives you all sorts of parameters, which are probably like the log likelihoods and stuff. Which if you're into that, you can look there.
Mm-hmm.
So
yeah.
All right, we'll move on. Move on. So the group level results. Okay, so that's how you analyze a single participant. Most studies have multiple participants, so we'll dig into how you would analyze your group level data. So this is a different tutorial. It's also available on the website. And we start off as always by importing our libraries. So I'll skip over that. And what we do is we define an analysis pipeline which we're going to apply to each person. So what I've done here is I've created a function called individual analysis, which will take in the path to the data and the ID of the person. So I just call them 1 through 5 because we've got 5 people here. Okay. And it's going to be the same analysis that we just had in the individual level tutorial. So we load the data, we convert to optical density, we apply the Beer-Lambert law, downsample to 7 Hertz. I don't know why it was 0.3. We separate out the short channels from the long channels. We'll make our design matrix, and then we'll add the average of the short channels to that design matrix. This is all the same as we just did. We run the GLM on the data and the design matrix. We'll define our left and our right regions of interest. And then we will export the channel level results, and we will export the region of interest results. And what we need, the extra steps that we need to do is, that's going to produce a data frame of results per person. We'll just, onto that data frame, we'll append the ID so that we can know who the data came from. And all the results up to now have been in mole. So that's always 10 to the negative 6. I find that annoying. So we're going to multiply everything by 10 to the 6. So it's in micromole, a bit more manageable. And this function will return the hemodynamic data, the region of interest data frame, and the channel level data. So the same things that we just saved in the CSV files before. So now what we're going to do is going to run that on 5 participants. I've stored the data here in the bids format, but you could just loop over your files as you would normally do. It's all NIRx data under the hood here today. So this is running now. It's going to apply that function to each participant. And that takes a few seconds, but that's finished now. So what it's done is it's read in the data, and it's created a long data frame of channel data and region of interest data. So we can have a look at the data frame, the channel data. So this is the same information we looked at before. We have the channels, we have the conditions. So for each source and detector, we have an estimate of the response. Was it significant? And what we've done is we've appended, now we have the different IDs because we've got 5 different people. So there's 12,000 rows here. And because it's got, you've got all of your conditions, and we've got our IDs from 1 through to 5. So that's quite a lot of data. Similarly, we have the same information for all participants now for the region of interest. So instead of by channel as above, we now have it by chroma and then condition and then region of interest. And what we've also done, sorry, one thing I forgot to point out is when you run the region of interest function, you can ask for extra demographic info. So NIRx stores the age of the person plus the gender. And so that can be useful as a in your second level analysis to include these pieces of information. There was a nice talk on this previously. So I thought I'd demonstrate how to do that. So if you include this, it will add these extra columns here with the age of the participant and the gender and their handedness, but I don't think that's stored in this data. So that's going to be all 0. So again, we get another quite a large data frame, and just as a demonstration of how you would if you want to cut it up differently, you can just query it. So you can say, give me all of the rows where it was HBO, or you can say, give me all of the rows where it was HBO and the condition was control, for example. So this is, and so if you ask for all of the HBO control conditions, we can see that for each person, we have a left and a right hemisphere estimate, and there's person 1 through to 5. All make sense so far? Yep. Okay. So looking at tables is confusing. So you can also plot this using ggplot. So if we just want to look at the control and the tapping condition for the region of interest data for each person, you would say from the group level results, I want on the x-axis the condition, on the y-axis the theta estimate, the color should be the region of interest, and the shape should be the region of interest as well. And so we can see that for each person, usually the control is sitting around 0, which is good. And we can already see that we're getting some sort of contralateral activity. So the right hemisphere looks like it's going to be bigger when they're tapping with the left hand, and the left hemisphere is bigger when they're tapping with the right hand. But we can see there's quite some variability in the amplitude across people. This is what I see quite commonly. And so what we're going to do is we're going to use a mixed effects model to analyze this group level data. So again, just to remind everyone what we're working with, we're going to take the region of interest data frame, which contains this information. And to start off with, we're just going to look at the conditions of control and the tapping left and right. And we're going to run a mixed effects model. So we're going to, this is really well summarized in all the Harvard papers, and they provide some nice examples. There's a nice table, one of the papers, that kind of works through this notation. And so we use the same notation in this toolbox. So we're going to ask to include fixed effects of region of interest, condition, and chroma, and have subject as a random effect. And if we run that, we get a nice summary of the statistical model. So we can see that for the right region of interest in the left hemisphere and the control condition in the HBO condition, the estimate coefficient is -0.2, and that it is not significantly different to 0. Similarly, for the right hemisphere, it's not. Whereas in the tapping conditions, we have some bigger coefficients and some smaller p-values. So this is how you would do your group level region of interest mixed effects analysis. Again, looking at tables can be quite boring, so it's nice to plot these things. So we have a convenient function called stats models to results, which will just split this cell. So if we run our model and we ask it to give us this stats model to results of the model, it just puts it in a nice data frame for us, which pulls out the information we want, like the region of interest and the condition. So this is the same information that was above, but I've just extracted the relevant pieces of information. And so we can now, again, use ggplot to plot that. So we'll have condition on the x-axis, the coefficient on the y-axis, and if it's significant, will be the color. We can see that. So this is our group level mixed effects model results for the control condition in both hemispheres, in both chromophore. That should be HB. Sorry, no, that's correct. Nothing is significant. Significant is false for all of these. And we can see that for the tapping on the left hand, the largest response was in the right hemisphere. So the contralateral hemisphere is expected. And in the tapping with the right hand, it was the left hemisphere that gave the largest response. So this is the region of interest group level analysis, or batch analysis, or pipeline analysis, different terms. Like somebody asked about covariates, and again, I would just recommend that you watch the previous NIRx talk on how to include covariates, but you can do it here. So I thought rather than, I would give a slightly different example. So let's say we now wanted to know if in the right region of interest, the tapping left and the tapping right conditions gave a different size response in the HBO signal. So we would take our region of interest data frame, we would only keep the tapping conditions, we would only keep the HBO signal, and we would just cut it down to the right hemisphere. And let's look at what that would look like. So we ran that. We would get a data frame that looks like this. So now we have the tapping left and right in the right region of interest. And we've got this extra factor here of gender. And so we could include that covariate in our mixed effects model, just as was described in the previous talk. You would go plus sex for this column here. And we would see that there is a difference between the tapping with the left hand and tapping with the right hand, that the tapping with the right hand in the right hemisphere is significantly smaller than tapping with the left hand, but that the gender has no significant effect on this. Now this is a data set of 5 people, so we probably don't take the exact numbers too seriously, but what I'm trying to demonstrate is just how to include covariates in the second level model. Any questions there, or can push on?
Cool. Yeah, all good.
Yep. So that was all sort of region of interest level analysis. It can, just like on the first individual level analysis, we can do the same kind of topographic plots as before. So it's quite long. I'm going to split this up. Just like before, sorry, excuse me while I clean this up. Now we're going to look at the channel level results. So we're going to take the channel data frame, and I'll show you that just to remind you what it looks like. So this is the input data. So instead of having regions of interest, we now have source detector pairs. And we're going to run a similar model. So we're just going to look at the tapping with the left and the right hand. And we're just going to look at the HBO signal for now. And we would run another mixed effects model. So just like before, we specify our mixed effects model, but instead of region of interest, we would specify channel name. So we would get the results of that. And now for every channel source detector pair, for every condition, we have a group level estimate of the response amplitude. And so, I mean, it's much easier to handle the region of interest data because it's more condensed. But so this is the individual level results, and it's too much to look at like this. So we can use, just like before, we can make these topographic plots. Excuse me. And so we can use this plot GLM group topo function. And we just want to plot the HBO signal in this example. So we pass it in the raw HBO signal. And then if we just want to plot on one plot for tapping with the left hand, we pass it in the tapping with the left hand part of this data frame. And we specify that we don't want a color bar, and that we can set the color map minimum and maximum. And so if we do it for the right hand, and then we do it for the HBR signal. So if we do that, then this is our group level GLM individual channel level results presented as a topographic plot. And we can see that just as before, when tapping with the left hand, you get a larger response on the right side. And when tapping with the right hand, you get a larger response on the left side. So that's, I think I will try and clean this code up in the coming months because there's a lot of arguments here that are always the same. So stick with us. So that's just like on the individual level, these plots, very similar. And similarly, we can also do the projection type plots too. So again, if we run this is the same code as above. We're just going to plot the HBO signal. We're doing the same mixed effects model. And we can use this plot GLM surface projection. And so if we do that, there you go. This is now the group level projection results. And so you can kind of change the color bar a bit too. And we can see that for this, when you were tapping with the right hand, that the response tended to be over this section of the brain, so in the left hemisphere. And then the last step, sometimes people want a nice tidy table of all of the channel level results. So let's say that your reviewer wanted to know for the tapping conditions and the control condition in the HBO signal, your channel per channel estimates of the response on a group level. Then you would run the mixed effects model just as before, where you specify your channel name, chroma, and the condition. And then you, as before, use the stat models to results to make a nice data frame. And then that would print out a table like this. So you would get for each channel, for each condition, you would get the coefficient, the p-value, the 95% confidence intervals, and if it was significantly different to 0. So I think this time I finished with 4 minutes to spare. So I did either just skipped over more things, or I was a bit more organized, one of the two.
We love the last one as well, Luke. So absolutely no problem. This was fantastic. We do have some questions, not on the second level, not too much. I think this is something that we have to try and then come up with questions later. But there are some general questions, could be from people who were not able to attend. If you wouldn't mind answering those for us. Yeah,
absolutely. And I think I've said before, but any follow-up questions, put them there. We've got a discourse forum for questions, or just log an issue. And I did just show a lot of things today without explaining the parameters. So if you go to the documentation and it's not clear, I mean, that's really helpful to let us know that it's not clear. So because if you can't understand it, then some of the other users can't understand it either. And so even just reporting, hey, I couldn't figure out how to do it, that's helpful because we can go back and improve the documentation.
Okay. There's a question. I think, I know you're going to fix this, but someone asked if they can have the link to the notebook again. But
yeah.
I think you need a couple of minutes to fix that, or?
Yeah, no, it might already be magically working. So if you go to the examples, and then if you go to any of these examples, so go to the group level waveform, for example, if you go to the bottom of the examples, you can either download the script, you can download the notebook, or you can launch this automatic notebook. There you go. And it will now start up a server in the cloud and allow you to run this code, and you can play with it and change it. And so this is probably the best way to try out MNE if you just want to play around. And I did, based on some other feedback on the website, there was always these instructions here, but I provided 2 alternative ways to give it a try as well. So I did listen to some feedback from the last talk. Yeah.
Thank you. All right. I think there's some very basic ones. I'll move to a bit more advanced ones to start with. Someone says, I will be interested to see blind source separation utilities and usability with multimodal data streams and advanced correction.
Yeah, that is more advanced. And so there is some blind source separation functionality. Like there's a lot of ICA type support in MNE. So I probably won't demo it, but I can show you where to find it. So if you go to MNE.tools, in here, within the documentation, within the tutorials, you should be able to find, so that would probably be under statistical analysis. No, where would I find ICA? Anyway, there you go. So there's sort of ICA or your different projections for cleaning up data here. So that might be handy.
Okay. Okay. This was, you mentioned this last time, but if you could give us a quick overview of the signal quality assessment tools.
Oh, yep. So that information would be on the MNE NIRs website. So if you go under the examples here, we can go through one of these. So there's a toolbox on signal quality evaluation, and there's I quite like the tools from the Samuel Montero Hernandez and Luca Polanini. So we have implemented both their scalp coupling index and their peak power algorithms. And so in this little example here, I sort of demonstrate how to calculate scalp coupling index either over the whole signal, or perhaps if you just wanted to calculate it over the first 30 seconds or the first 10 seconds, you would, I've demonstrated how to do that. So you would crop the signal first and then calculate it. And then if you set a channel as bad, it will be removed from the downstream processing. And anytime you plot the data, so if you were to plot the sensors that have been marked as bad will be marked as red. So that's kind of handy to sort of know, is it always your sensors on the left side of the head that are bad? And is that because, you can't be bothered reaching over the person to sort of wiggle them before your experiment, or to, sort of look for some patterns. So I find it quite handy to visualize the bad channels. And there's also the peak power metric here too, which is very handy as well.
Okay, yeah. And of course, there's the more standard tools like coefficient of variation. that uses.
Yeah, and if you're doing a waveform analysis, you can just reject on peak-to-peak threshold, so you can say anything with a peak-to-peak value greater than this, just exclude. So those sorts of things are in there.
Okay. Not so much about analysis. Is batch processing possible? And there's a follow-up. Is transformation of near site optode positions, which is the NIRx montage design software, to cortical MNI-based optode positions possible?
Yes, so the batch processing, so the way I do that is, I define a function for each individual, and like we did with the GLM. So it's it would be a similar pattern if you did it for the waveform analysis. And I would just share here. So this is the group level waveform analysis example. And I will sort of go through it because it's interesting. So it's very similar to the GLM. So you define this individual level analysis, and that will return your epochs per person. And this sort of, all this code here is exactly what we went through in the last webinar. So you just sort of package up that individual level analysis into a single function. And then once that's done, you can use these like plot, compare, evoked signal responses to make these group level waveform analysis. So here the ribbons or the error bars represent the 95% confidence interval across participants. And that's and you can do that either all the channels. So in this plot here, you can see this was done over all the channels, but you can also generate regions of interest like the GLM, and you can create these plots per region of interest too. So you can see this is the top row is the left hemisphere and the bottom row is the right hemisphere. So that's how you would do batch processing on a waveform analysis.
Okay. This was also covered by you in the last talk. So if you could also give us a brief, let's say, revision of it. Any hints on visualizing each step of the processing chain to see effects of parameter changes on output?
Yeah, so for that one, I like to run after each, we'll go back to the code. So where is, so we'll go to the individual level code. Bear with me. So if we reload the data again, what I like to do is, here we go. So we'll start off with the raw intensity data. And I like to plot that, for example, and then after each step, I just run that plot step again. So then I would run this optical density, and then I would run the plot again, and you can see the differences. It's particularly evident when you run something like the TDDR. There's an example in here of the artifact correction. So this is what I should have shown you. So this is the dot plot function. So this is just your standard plot of the data. And so this is like the before. And then what I do in this example is I add some artificial spikes and baseline movements to the data. So think of this as the before. So we plot, and then I apply this temporal derivative distribution repair algorithm, and you can look at the after. And so this is how I sort of process and then look at the data after each step, and you can see the before, there's this sort of baseline shift here, and the after, that baseline shift is gone, and you're just left with this little kind of brief artifact.
Okay, right. I think that's it. This was extremely helpful and extremely interesting, of course. And judging from the responses, and there are many compliments for you, Rob. A lot of people saying thanks. And the resources are amazing. And of course, I think more people will come back to you once they do some hands-on.
Right. It sounds like the most trouble people have is installing it. So if you if you you know, just don't get stuck on the first hurdle. Just reach out if you get stuck there.
Absolutely. Okay. Thanks so much again, Rob. I know it's super late in the night for you, and we'll have to let you go at some point. But
thanks very much. Thanks for having me.
Absolutely. The users can reach out to Rob directly or write in the forum, which he likes very much. And I have seen that you get very frequent replies, which is amazing. You could, of course, always reach out to us as well if you have any questions specific to our devices. And yeah, thanks so much. I hope you enjoyed, and we'll see each other next time.